All right, so there's not just one heavy variable, right? There's 40 of these. Holy crap, there's 25 of those. There's 6 of those. So what's that math add up to? A lot. Somebody has to do it. So 4 times 25 is 100, so that gives us 1,000. 1,000 times 6, 6,000. 6,000 heavy chains. How many light chains? Total?
320.
320. What's the total diversity you could have? It's whatever those two numbers are multiplied, which is what? 6, 12, 18, 19, 190,000? Am I right? I'm sure it's wrong. 3 times 2 is 192,000. Was that close?
1,920,000.
1,920,000. I was close. I was only off by a decimal.
Only up by a million or so.
Only by. All right. So that's just with combinational diversity. That doesn't have anything to do with the complementarity determining regions, which I don't even know that you can calculate that. And there's going to be more diversity when we start slicing and dicing them together. This is good, yes? Because we want to be able to recognize every single thing that is not you. Okay, there's a lot of you. How much of you is there? How many cells? It's 3 times 10 to the 9th. All right. Well, and that's cells. Cells are huge. So we're talking epitopes. You need to recognize everything. All right. We're going fast. I feel as though your head might spin off because I'm going to keep going. Because this is the best part. Everything's the best part. I really can't, I mean, every single thing is the best part. How and why does rearrangement occur? We can't really know why. We know how. So there are these sequences of DNA called recombination signal sequences. All right. Obviously, we've named them this. Okay? They don't come like stamped where it says recombination signal sequences. RSS. And down here, they're showing you an RSS. Each RSS has 3 things in it. It has a heptamer. What's a heptamer? How many's hep? 7. All right. What's a nonomer? A 9. And a spacer. And in this case, they're showing you a spacer that's 23. And over here, this is another RSS. Notice it has a heptamer and a nonomer, and the spacer over here is 12. So there's a rule, the 23-12 rule. Okay, the spacers can only be 23 or 12. Tell me something you know about DNA. Anything. It's double-stranded. Good. What else? Thank you, Brad. It's helical. Okay. These are sequences of DNA that are going to be recognized as places for enzymes to come and sit down on. Why would we have a difference in spacers, 12 and 23? What's that? Different enzymes is a good idea, but not right. Okay, so it's a helix. Okay, if you're a helix, if I'm a helix, pretend there's like a rope going around me. Now that would be really fun. Let's tie me up in a rope. Well, I'm either going to recognize these 2 strands of DNA and bind here, or I'm going to recognize the next one up and bind there. 12 is the short one, 23 is the next one. You know what I'm talking about? Who doesn't know what I'm talking about? It's not the major and minor group. It just has to do with the DNA is in a helix like a ladder, like a stairwell. And right, I can't, if I need to bind, I'm not going to bind to something that's here and then on the back. Right? I can't bind here and on the back. So what I do is I can bind to two things that are right here, but they're not together. They're separated by sequence, either 12 base pairs or 23 base pairs. Okay. We'll come back to that. Okay. So we're talking about one or two turns of the DNA. And these sequences are found right next to the pieces that we're going to put together. It's either going to be VJ in the light chains or VDNJ in the heavy chains. So you have these recombination sequences that allow us to start putting these pieces together. I'm losing people. I see you in the same direction. Here's the loop-de-loop. Okay, you will not understand this today. I highly recommend that you make your own. Okay, I couldn't get it until I made something all by myself, and I proved to myself, oh yeah, they're going in opposite directions, but when I do the loop-de-loop, it's so stupid, right? I mean, you feel stupid doing it, but it's the only way. I need to see things to believe it. I mean, I could memorize it, but I wanted to understand it. All right, so here, they're saying take this, right, going in this direction, the VN and the J. The only way, right, you can get them, all right, you can't just loop it over here. You have to go loop and around. So these have to point in the same direction so that these are pointing in opposite directions so that when you put them together, okay, that part you won't have trouble with. The part will be, well, where the fuck does this go? What? Where the hell does that go, right? So if I'm going to put this together with this, the red goes with the purple. Here's the purple. Here's the red. And then all of this comes. So you get to this red, to this red, that then attached to this yellow. All right? And then here. So if I were to draw something in here for you to see that, let's just say this is the striped part. That striped part is here. All right, I see your faces and you're disbelieving, and I totally understand that. It is unbelievable. Okay, so we'll come back and we'll start here again and we'll talk about the enzymes that do this. But this is what's creating additional diversity and specificity, is how they join together. Okay? It's Monday. That was hard. Let's take until quarter after. Okay? Let's take until quarter after, and we have a relatively short experiment. I'm recording. I don't know if I'm recording over. I don't know what I'm doing. All right. Did anybody go home and try this at home? No. All right, well, still have to wrap your head around it. The big difference between just making the individual loop, all right, in the individual loop you created something we called, the whole DNA was called a signal joint. And that signal joint will get degraded, and so you've actually lost part of the genome. In this case, we've actually just flipped it around a bit, right? So you don't lose anything because all of this blue stuff is still here, and then we have the red stuff. And so we haven't lost anything, we've just flipped it around. And I guess the big difference would just be, if you looked at that DNA, where would cut sites be, right? It could be that you've lost the cut site or you've moved it and you made pieces smaller. So if you were to look at a southern blot, it would just change the way things would get cut. Now here is the mechanism. Remember I told you we would get to how this happens. That was that was the what of what happens, and this is the how it happens. So these are a bunch of enzymes that are going to act to do somatic recombination. And the first ones are cleverly called RAG 1 and RAG 2. So now if somebody says you're on the RAG, you can say, well, what are you exactly talking about? All right, this is, right, recombination, all right, and these RAGs are the first of the enzymes that come in. They will recognize the RSS. The RSSs were the sequences, the heptamer, nonamer, and spacers. And they will bind to the sequence of DNA right where, so in this case, they're showing you a loop as opposed to a loop-de-loop. All right? So the blue and purple are the RAGs. So RAG 1 and 2 come in, and they cleave the DNA. So they are what kind of an enzyme are they? Lisa knows. All right. If they're cleaving proteins, what are they? Proteases. And if they're cleaving DNA? Deaminases. They're called nucleases. So, right, you've heard of things that cleave RNA, RNases, all right? I mean, this could be called a DNase, all right? But the general term is a, whoopsie, let me grab a pen here, is a nuclease. So they have to bind the RSS, they cleave at the RSS, and what happens, which is really cool, maybe not for you, but for me, is that on the ends of these DNA, where we had, in this case, we're just doing a VJ joining, so this is a light chain. At the very ends here, the DNA creates this closed little loop called a hairpin loop. Does anybody have any idea why that might happen? This is your organic chemistry coming back to haunt you. Some of the only words that I actually remember from organic chemistry that I don't know what they mean because I failed it.
From the hydrogen bonds, the hydrogen bond interactions.
It's going to create a covalent bond. And so, how do we do that in OChem? Nucleophilic attack. Right? Does those words ring a bell? Yes? You don't have to know either, because I still don't know, and I still don't give a shit. Okay? I really don't know. Fuck. I hated OChem. I hated it with a passion. Something happens, and it creates this little hairpin loop. So these, man, you can't hardly see that, can you? Hairpin loops. All right, that's way better for me to see. I'm like, ooh, I can't see that either. It's not the RAGs that do that. It just happens naturally because of whatever the ends are. Nucleophilic attack, blah. That's going to matter because we have to go back and we have to cut them later. All right, another interesting feature of this is that there are proteins that come in called Q70 and Q80. And maybe not interesting at all for you, but interesting for me because in my previous life, way back when, and it wasn't that long ago, when I was doing my PhD, I worked for a guy that is endocrinologist. Endocrinologist? Rheumatologist, sorry. So the other guy was an endocrinologist. A rheumatologist. What do rheumatologists do? What's that? Autoimmune stuff. And so one of the things he was obviously interested in C-reactive protein, as far as it plays a role in the immune response and whether it has a role in autoimmunity or not. But coups, so these are proteins. Coup proteins are known to have autoantibodies to them in some autoimmune diseases. And so what's interesting to me about this is, oh look, I had done some work in his lab and we had talked about coups. I had no idea what they were, where they played a role. And now I can actually find a role for them. What they do is they come in and they hold together these two strands, right? So the two strands that are going to make up the signal joint and the two strands that are going to hold together what we call the coding joints. They're called coding joints because they're going to actually flip together and make the protein. So the coups are basically holding things together. Why do you have to hold things together? What held them together before? I mean, this was just DNA, right? And it had this loop. And so they couldn't get too far away from one another, but once you cut this off, this whole thing could just float away from each other. And if they float away from each other, you're never going to get them back together. It just can't happen. So they have to be held together. These guys, they could probably end up, because they're on a shorter strand of DNA, they could end up interacting with one another. But these guys, no way. You just cut a chromosome. Okay? And I think that's it. So if we want to think about making a list of proteins and what they do and what roles they play, that would be a good exam question. How very subtle or not. Okay. Interesting feature of the RAGs is that they're only expressed in developing lymphocytes. What are the lymphocytes? B cells and T cells. Just B cells and T cells. So these genes only get turned on during development in B cells and T cells. Yet another reason why we don't see somatic recombination happening in cells other than B cells and T cells. Somatic recombination doesn't happen in kidney cells. Doesn't happen in heart cells. Cleavage matters. And the cleavage sites are based on the RSSs, right? And these have nuclease activity. If we do express the RAGs in other tissues, if you could turn them on, you would start seeing some of this happen. All right? So it's really a matter of how do they get turned on only in these cells? What turns them on? And those are going to be transcription factors. RAGs, all right, and then I remember putting this slide in because people wanted a list of the different enzymes. So you have the COO 70 and 80. There's a DNA protein kinase. Artemis. I don't know why they called it Artemis. Who was Artemis in the... He was a Greek mythology, I'm pretty sure.
Huntress.
What's that?
She was a huntress. She was a virgin huntress.
She was a huntress? Artemis is a woman's name? What the hell kind of...
Artemis and Diana are the same.
Ah,
Artemis
interesting.
is Greek, Diana is Roman.
Well, you're going to see Artemis has this nuclease activity as well. And we're going to use DNA ligase 4 along with XRCC4. And these two are going to help put DNA back together. So we're actually going to go through this whole process. RAGs, COOs. Okay, no problem there. The next thing that happens in the signal joint is you have the COOs holding it together and the DNA ligase and XRCC4 come in and ligate the two ends of the DNA. And that creates this circular DNA, which I told you, since it doesn't have an origin of replication, it will get degraded eventually over time.
cell undergo mitosis after the recombination?
So cells will undergo mitosis. So one of the best things about, right, you're creating one, let's say this is a B cell, right, because we're talking about heavy and light chains. This is one B cell, and it's going to express one antibody with one heavy chain and one light chain. And for that to be effective, let's say you get the flu and this one happens to be the good one, it has to undergo mitosis because you want to make a lot of antibody. So there's tons and tons of mitosis that occurs after the fact. What's that?
So you're cutting pieces out.
Yeah. So these cells,
they're becoming more and more specific. Just
once you already have a B cell receptor, and that cell expresses that B cell receptor. So this is only during development. Okay. And what happens is B cells, this can happen through the course of your lifetime. T cells, not so much. And we'll talk about why and where this all happens. All right, well, those signal joints, remember, they look like this. So they had the little arrows, and they had very precise ends on them. And they literally get put together, this end to that end. That's all it is creating, what bond is it making, the ligase? Speak up. Felix? A phosphodiester bond. All right, that's all it is. It's creating a phosphodiester bond so that these two are ligated. There's no funny business in between here. Okay? Over here, we're going to have funny business. So we say that joining of these is very precise. It goes like this. Dink. Just like that. Funny business means we can actually add some DNA to these strands that wasn't there in the first place. You're like, what? It gets better as we go. This is like the coolest story. All right, here we are holding together with Q70 and 80. Along come DNA, PK, and Artemis. All right, DNA, PK. What does DNA, PK do? Phosphorylates. Just think again. So, what other protein is there? Artemis. So DNA PK is going to phosphorylate Artemis. What's that do to Artemis? What's that? Changes her conformation, which could do what? Artemis is a nuclease.
Opens the DNA?
She is going to open the DNA, now that we know she's a she. All right? So what does the phosphorylating do? It activates her. Before she's phosphorylated, she can't cut open the DNA. After she's phosphorylated, now she can open the DNA. Why not just have Artemis floating around all the time, ready to go? You would just cut open DNA indiscriminately at bad places. By linking these two together, this has to bind to the complex, which allows for the phosphorylation that activates Artemis to cut open the little tiny things you can't see. Okay? So here are our hairpins. Our hairpins are open. Okay, we just closed the hairpins. Yes? Cut, close. Cut, open. You may say, why are we doing this? All right, and what's the answer? We don't know why. All right, we don't know why. We know this works. The next part of this cascade of events is something called TDT, terminal deoxynucleotidyltransferase. What? Terminal means at the end, okay? Not terminal as in dead. Terminal is at the end. What's a deoxynucleotide? A, C, G, or T, as opposed to a ribonucleotide. Transfer. It's going to add on bases, right, nucleotides, to the ends of these opened hairpins. And you should be saying, what the fuck is going on? We're not only messing around with this DNA and cutting some pieces out, now we're going to add back in DNA randomly. I love this. Oh my God. You know why I love it? Because it's a crapshoot. What does that mean? It's just throwing the dice. And you throw the dice, you throw some nucleotides in there, and you hope that those nucleotides are going to generate a protein that's going to be functional. All right? We're going to go into this in a little more detail. After nucleotides get added, then the DNA ligase 4 and XRCC4 come in and do ligation. So all we've done is we've joined the V region with the J region. That's all we did. Right? It's a little more complex than just lining them up and flipping them around. Right? By adding in extra nucleotides, we're increasing the diversity in the heavy and light chains. Right? It's random. Anything could show up there. What else could happen? If you're adding random nucleotides, what could happen? Remember the two pieces that have to line up, right? When they line up and we do transcription, these two have to create a functional protein. So if you randomly add nucleotides, you could add nucleotides that are not in triplets, and you could cause a premature stop or right, frame shift mutations. It seems a little dicey. Crapshoot, dicey. See, it's all coming together. Well, you'll see why. It's all good. So this is just a recap. The rags line to the 23 and 12 spacers and cut. These loops are formed because of the nucleophilic attack. The coups come in and hold everything together. DNA PK phosphorylates Artemis. Artemis is a nuclease, and it's interesting because it only causes one nick in the DNA on one side. So it only opens, it doesn't cut the whole thing off. It just opens one side. And then that TDT comes in and does crazy stuff. So that's what we're going to talk a little bit more about is the TDT. All right. The sequence of DNA that gets opened up, all right, they're showing you this as these are the little pieces of DNA that are sticking up. Here's the D and here's the J, and right, we're going to join them. Well, it cuts, and even though it looked like it cut off exactly at the top, right, so it looked like this, I don't know. Even though, where am I? even though it looks like it cuts everything off here, there's still something there. All right, there's still some DNA there. Their pictures can't be perfect. So this is the DNA that's still stuck to the D, and here's the DNA that's stuck to the J. All right? It gets cut, and there's a couple nucleotides still remaining, and you see the hairpin. And then when the Artemis cuts, it only cuts on one side. So now we have just a handful of nucleotides. I don't really know if it's 2 or 4 or 6. Okay, I don't think it matters that much. But what matters is these are the DNA. So this DNA was not ever in the original chromosome. And the reason it was never in the original chromosome because they were on different strands. So immediately, right, it says, is that a C? I think it's TC, right? And it goes TCGA. There was never a TCGA. It was TCCA. So, right, this got cut out. Bam, this got cut out. And when it flips over, you immediately no longer have the same sequence that was in the original chromosome. Okay, we call these sequences of DNA, this 4 here, it's called the palindromic sequence, P, right, for palindrome. Why was it a palindrome? Same forwards and backwards, okay? And then the craziness begins. Oh, how I love crazy. The TDT can add up to 20 nucleotides of, right, non-genomic DNA. This is just random shit. Just adding some nucleotides. We don't care. And it doesn't matter what you add on this side and what you add on that side. Just throw some nucleotides in. And we call that, so you end up with P and N nucleotides. Nucleotides that are from the palindrome, and then nucleotides that are brand new.
Just until they can stick together?
Just until, all right, so we add a whole bunch, and then it gets even more fun, because they're not going to work perfectly. And so, it'll add to some point where it will create some overlap. And as soon as they can stick together, then they'll remove some. They'll get rid of the ones that are not complementary to one another. And now we have DNA that has the ability to base pair with one another. And these nucleotides will get filled in by an enzyme. I'm trying to think. It says, I believe it's the DNA ligase 4 and XRCC4 come in and repair this DNA. It can repair it because it has a template now. Right? So it can add nucleotides because it has a template. But you end up with nucleotides that are part of the palindrome, totally brand new nucleotides, and then the other side of the palindrome. This sequence of DNA was never in your genome before. That's cool stuff. I love that. Even if it is DNA, right? I don't like DNA much, but I do like this. So the idea here is that we're creating variability in how we join the B and the J region. So exactly the same thing would happen for the heavy chain. When we link the D with the J, there's going to be exactly the same process. And then when you link the V with the DJ, exactly the same process for the heavy chain. Same enzymes, same process. So we're creating more diversity and ultimately greater specificity, the possibility for greater specificity. You are also creating this possibility that you're making shit. Turns out your immune system doesn't care. It's willing to take the chance that it's going to mess up a receptor. It's willing to do that in the hopes that it's creating enough diversity otherwise to make up for that energy lost. So we call that, I think it's called joint diversity. It's not joints like smoking a joint. It's joint diversity where they're actually joining the pieces together. Okay? All right. What happens when your enzymes don't work properly? So, did anybody ever see the movie The Boy in the Bubble? Right, based on a true story. Okay, if you want to see a sad story, watch The Boy in the Bubble. All right, it's a really sad story. This kid was born with a defect in adenosine deaminase, and the parents were convinced by the scientific community that they should keep their son in this bubble environment until they could make a therapy that would fix his problem. He had a genetic defect in this adenosine deaminase. Now you might wonder why adenosine deaminase matters. Okay. So adenosine deaminase, here's adenosine, right? We're talking about nucleotides that have an A on them, right? This is the non-toxic substance. Is this toxic? No, this one's toxic. And if you have the adenosine deaminase, it takes off this amine group and generates deoxyinosine. So some of you may have seen inosine before. And this is not toxic. And what's interesting about this is that this toxicity is specific for the bone marrow. And it goes into the bone marrow and it ends up killing your B cells. So if you don't have this adenosine deaminase, you end up not making B cells properly. And now you can't fight off, actually you can't fight off T cells and B cells. And you can't do somatic recombination. So he ended up having, his name was David Vedder, and he had severe combined immunodeficiency, and he had to live in the bubble because literally everything could kill him. Right? Breathing the air could kill him. And it was a terrible life. It was absolutely awful. He escaped from the bubble occasionally, got very sick. It's this really sad story. This is the first time we really started talking about doing gene therapy. Didn't work. But not everything great works the first time. So it is a sad story, but there are children that are born with these types of disease. So this child has a disease called Oman syndrome. And I don't even remember what this one is. I think it's lack of T cells. And I don't even remember which enzyme. This is circulating B cells. Yes. And so what happens is, because they don't have B cells, they end up with T cells that go into their skin, and their skin ends up with all of these little, I don't know what you'd call them. They're not pock marks, they're like bumps.
Blisters.
What's it?
Blisters?
Blisters. And it's just a nasty, nasty disease. Any severe combined immunodeficiency is going to be bad. What's the one you guys know? AIDS, right? And that's just, why is AIDS a immunodeficiency? What are they missing? Really? I mean, there's a gajillion movies that you could watch to find that, right? What's the one with the The guy got really skinny. No, not Philip. No. More recent. He does the car commercials now. I can't stand Matthew McConaughey. I can't stand him. Dallas Buyers Club.
Yeah.
That was a good movie. I had no idea that was based on historical facts. If you haven't seen it, you really should. And I get all my history through movies now. All right. So in HIV, HIV targets the T4 cells, right? CD4 positive T cells, so you don't have those. CD4 positive T cells are required to help B cells become plasma cells. So now you don't have CD4 positive T cells, they don't help the B cell cells don't make antibodies, and you're susceptible to all sorts of things. Okay. So this one's fatal. All right. Not good. We can do a lot with transplants now, but obviously not enough. So there's lots of ways to create diversity. We talked about combinational diversity, combining heavy with light, but there's two different lights, lambda and kappa. There's now junctional diversity, and that junction is based on TDT, when we're joining V with J regions or D with J regions. All right, and separate completely from this, because we do love to just make things even more fun, so I'm just going to make a line here that shows you. This is separate. We haven't finished what you're going to do with an antibody. There's something later on that we can do called somatic hypermutation. So you make a B cell receptor, and after the fact, after you see an antigen, we can actually take that B cell receptor and we can tweak it a little more, trying to make it even higher affinity. Okay? Does anybody have any questions about that? All crystal clear. Okay. Without your notes, right? Just close whatever you have, take a new page, right? Blank page. See if you can at least list the enzymes in order, and work together if you need to, but list the enzymes that do this somatic hypermutation, somatic recombination. Just try it. What's the worst that'll happen? You'll get it wrong. And if you can't list them in order, see if you can just list them. If you have to, draw yourself the picture. Give it a go. But Hebrew, you probably need to get some more sleep.